Okay, great. So I've called this Pharmaceutical Escapades, and this is the work of my PhD student, Cecilia Ojimaye. I'm the leader of the Environmental and Nano Sciences group at the University of the Western Cape, and several of my students are involved in this kind of research. As you know, chemical pollution is becoming a greater and greater problem. And when you look at the case of drinking water in industrialized and populated states, it's actually contaminated with chemicals, and South Africa is not escaping this problem. We know from the data that my colleagues have collected that we can see how much of these endocrine disrupting chemicals are being imported and exported over the last couple of years. So we are really increasing the imports of these compounds a lot. The reasons why these pharmaceuticals escape is because of the way that we live. And here is a kind of a life cycle of the whole process. It's a bit small, but I think if you would like to, you can take note of the website that we've created, which is called waterstories.co.za. And I would suggest that you go and look there if you want to see more details. But essentially, these compounds don't stop moving just because you've taken them in your body. And one of the main routes for the escape of these compounds is our wastewater treatment plants. And here you see the national green drop results for 2011. And you can see that in the country, there were only about 5% of our wastewater treatment plants that were optimally working. The rest were in a poor to abysmal state, and the situation is not getting any better. Here in the Western Cape, we have a whole lot of wastewater treatment plants. If you can have a look, we've got these marine outfalls at GREENPOINT and at CampsBay and at Hout Bay. And then there are these wastewater treatment plants here on the Cape Flats. And you can see what volumes are being treated in those wastewater treatment plants per day. These are megaliters that are going out into the environment daily. Now, when you look at False Bay, the city has provided figures showing that the coastline is extremely contaminated with E. coli and Enterococcus bacteria. So it's clear that the sewage is flowing back to the coast. This is a graph that shows you the contamination from Miller's Point all the way to Kwiggle Bay. And you can see here at Makassar and at Strand, the beaches are very contaminated. But this E. coli is not the only problem. The problem is all the chemicals that come with the sewage. So here is a graphic of all the different types of emerging contaminants, but today we're only going to focus on these pharmaceuticals here, such as antibiotics and antidiabetics and antiepileptics, etc. So why are we worried about these compounds? Their impacts are quite well documented, and they cause things like feminization of the population and downward trends in semen quality, and that results in declining sex ratios with more females than males. It also causes sexual abnormalities and developmental abnormalities and reproductive impairments, as well as disruption of your endocrine system and persistent antibiotic resistance, etc. So it's not a trivial problem. And here is one of the papers that was recently published showing you about the antibiotic resistance in river and stormwater in the Western Cape, and the levels of antibiotic resistance are very scary. Our study sites were around False Bay and at GREENPOINT and CampsBay. So GREENPOINT, CampsBay, we've also started working on Hout Bay, and then at these points around the bay. So there were eight sites around the bay where we did sampling. We looked at these compounds. So we looked at perfluorinated compounds, industrial compounds, and then a suite of pharmaceutical compounds. And we analyzed for these compounds in a variety of organisms. So we had a look at limpets and at mussels and at starfish and at sea urchins and top shells and seaweeds. So on the GREENPOINT study here you can see the marine outfall. This is a photograph by Jean Trespan, and that's not muck released by that ship. It's actually coming up from the marine outfall. The sewage is pumped straight to sea without treatment. And here you can see sometimes that sludge comes onto land when there's a storm. You can see up there, there's a footprint of somebody stepping in that sludge between the cars that were parked on the pavement there. And you can see the color of the water here where the sludge is pushed up against the coastline. And when you test that water, you can see that we have a whole host of these pharmaceuticals and personal care products in the seawater. We tested about 8 different sites there. And if you have a look at the y-axis, the nanograms per liter are sub-nanogram per liter in the seawater. So it's relatively low concentrations, but you'll see there's a little bit of all these compounds in every water sample that we took. Then we had a look at the marine biota, and if you'll notice here that the compounds are actually up concentrated by about 10-fold, and you can see that the mussels, the limpets, the sea urchins, the starfish, the shells, and the seaweed actually had these compounds in them, and quite a lot of the different compounds compared to the seawater. We went along to CampsBay and we had a look at what is in the water there, and you can see the marine outfall marked with an X there. And you can see this is a bad day when the sewage has actually been pushed against the coastline. So much for blue flag status on that CampsBay beach. And here you can see the pharmaceuticals in the marine biota, and it's heading up. This is more concentrated than what we saw at GREENPOINT. And you can see here, for instance, the compound diclofenac is quite prevalent, and so is phenytoin and sulfamethoxazone, which is an antibiotic. Then we had a look at False-Bay. I'm skipping through the results so that we can get through the talk quickly, but we did those 8 sites in False-Bay. And here you can see in False-Bay, all these sites, seawater was actually contaminated. And you can see how much of the antibiotic sulfamethoxazole was there and the diclofenac in the seawater. Now, when you think about that, nanograms per liter in millions of liters of water, and you need to back calculate that to think of the tonnages that are going into the sea every day based on the nanograms per liter that we're testing in the seawater. We also looked at the sediments. And here you can see that compounds like diclofenac are accumulating in the sediments quite strongly. So when your kids play on the beach sand, they're probably getting some microbes as well as some chemicals into their little bodies. And here are the biota that we looked at. So the limpets were present at all the 8 sites. Mussels were present at a few of the sites. The sea snails were present at a few of the sites, etc. And you can once again see that diclofenac is one of our biggest problems, and the levels are pretty high here. We're talking about nanograms per gram of dry weight. So here it's not per liter anymore, it's nanograms per gram dry weight. And each of these creatures had all the compounds that we tested for present in their little bodies. And here's the seaweeds. Now people are harvesting the seaweeds for food, and so are they harvesting the mussels. And those are full of these compounds as well. And once again, you can see how much diclofenac and sulfamethoxazole are present in the seaweeds. We did the risk assessment, and you can see that as far as acute toxicity is concerned, the levels were not acutely toxic, but when we start looking at chronic toxicity, these marine organisms are being exposed to a chronically toxic environment, which will have effects on them as time goes by. We also did a study on fish, which we bought at the Kalk Bay Harbor, and we found that these fish actually had these compounds in even higher levels than in the marine biota, and you're very welcome to read our papers on this. which is worrying because the fish comes onto your plate as food. We also found a whole host of herbicides in those fish. Now, we also did an analysis where we didn't quantify, but we qualified, and we found up to 1,000 other compounds in these marine organisms. We couldn't quantify them, but we could identify them. So actually, each of these marine biota and fish are actually having to deal with a total onslaught every day of different chemicals. And this is really showing us that our wastewater treatment plants are completely inadequate. So the conclusions of our study is that sewage flows back to shore in detectable quantities that could be harmful. And there's measurable presence of these selected endocrine disrupting chemicals in seawater sediments and in many different species. The compounds are present in the organism's tissues at concentrations much higher than in the ocean. These compounds thus bioaccumulate in marine organisms. And because of that, we could only test for a certain amount of them, but we detected many others there. And we don't know what these combined effects and concentrations will have on our marine biota. And we are talking about the blue economy, but we're busy killing our blue economy because it's busy contaminating the fish stock that is wild caught in the marine environment. And we've demonstrated the bioaccumulation up the trophic level. And essentially the bioaccumulation is due to the molecules' hydrophobicity, their molecular mass, the number of hydrogen bonds they're having, their polar surface areas, and their rotatable bonds. This is actually known as the Lipinski's rule of 5. And this is what actually causes the bioaccumulation. And the risk obviously depends on the concentration, the exposure time, the volume, and the complexity of the mixture, the organism type, and the metabolism rate. So it's very difficult to actually properly estimate that risk because we were just calculating it based on the type of calculations that are available in literature. So finally, I would just like to thank our sponsors for this study and thank you for listening, and you may ask me questions if you'd like.
So Prof. Petrick, I must congratulate you. When I saw 32 slides, I was like, oh my. But you actually did this under 15 minutes. And it's a very interesting story that you tell. Do we have any comments or questions? Because the question that I had is, so what was your analytical, were you using LCMS for this? Because I guess the quantities are very small.
Yes, LCMS. And okay. Yeah, we had to find a high resolution instrument because most instruments are too, the resolution is too poor to actually measure.
Yeah, sensitivity is an issue. And then over how many years did you do this, and was it consistently the same? Was there seasonal variation, or maybe that didn't come into this study?
Well, because of the extreme problems of getting these compounds analyzed, we couldn't really do several seasons. We would have liked to, but we really need to start thinking about how we're going to stop this mess, because I think we've now shown that these compounds are everywhere and our wastewater treatment plants are completely inadequate for removing these compounds because they are finding their way into the open environment. And pharmaceuticals can only come from people's purification, when we're talking about what ends up in the sea.
Yeah, okay. Well, Teboho, and then I want Eike to just make a comment around analytical. Eike, if you can. Is it possible to use HPTLC at least for screening purposes or something? But let me take a question or comment first, and I'll come to you, Eike. Yep. Teboho?
Oh, thank you, Prof. Oh, no, I think she partly answered my question, because I was wondering that, okay, with all these drugs being present in water, are we only concentrating on the incorrect disposal, or we are also concentrating on the consumption of those drugs by patients? And some of those drugs, they are also renally excreted unchanged, which would be like...
Yes, this is exactly This is exactly the problem. You know, our pharmacists were completely horrified when they saw how much unmetabolized drug is getting through the wastewater treatment plant. And that is basically because a lot of these drugs are not fully bioavailable, especially when you're talking about generic drugs. They often are less bioavailable than the original drug because of the way that they're compounded. And we didn't even start looking at the metabolites because remember, a proportion of the drug might be metabolized to the active component in the body. And we didn't even start looking at those metabolites. So all we're seeing at this point is the unmetabolized drugs. So those levels are actually lower than what one would actually find if you went to go look for the metabolites.
Okay, thank you, Aika. Can you come to our rescue? Can we have really affordable analytical instrumentation?
Well, yes and no, but the question was asked correctly. The levels are pretty low, although they are pretty high. But if you want to take the samples as they are, then you would need to go with some really sensitive instruments. And what we have seen here was certainly a targeted analysis for a set of samples. If you would concentrate the water, which is really easy, you just have to evaporate this, then your concentration get much higher, and then you can do this. But this is an extra step. Ideally,
Yes,
the online injection would be the way to go if that's possible. But this is screening for the known. I think there is a lot of work being done in the past for untargeted screening. That is effect-directed analysis, where we are using microorganisms, yeast cells or bacteria that have some action or interaction with those pollutants, and then you can search for estrogenicity, for example. You can look for antibiotics, for example, and then you get just a presence, and you can screen for the cleanliness of water. And this is what several wastewater institutes are doing in Germany, for example. They are screening their water by HPLC, basically. And if there's any positive response in any of those 5 tests that they're employing, then, well, we have to find out what this is, and then we are analyzing. I think this is it's already very late in the development now that we are finding it in the oceans and we are finding it in the coastal waters. The question that was asked, what can we do at the earlier stage? So this is the wastewater treatment. Can we do before and after and see what happens? So okay. HPLC can do but so much. The levels are pretty low, but most of the time after concentration, we can see this. Effect-directed analysis would be the ticket. We are going for the unknown, and we just figure out what is of biological risk in here.
Yeah, so I guess they inject, the dilute and inject or inject method, you'd have to concentrate. But maybe it's, it's worth having such an instrument. Prof. Petrick, last words on that one?
Well, we did up concentrate by using SPE cartridges.
Oh, yes.
So that is one, we did that extra step. So everything that we analyzed had a pre-concentration step to get it into the range of the instrument. But even so, the instrument's resolutions generally are very low. So it's difficult to quantify. But yes, one can qualify. You can identify the compounds, but not so easily quantify unless you've got the standards and you can draw calibration curves up. So for all those analytes that we checked, we did have calibration standards, and we had deuterated standards so that we were able to look at recoveries and see what the limits of detections of the instruments were.
Okay,
so
thank you very much. It's going to be a problem. It would be interesting to see what Durban presents, for instance. I think it might even be much worse. But then you talked about upstream. So what is it that we can actually do? And this is actually what happens in the UK. So I'm going to try and play this. And our good friend Abel is a pharmacist. He owns the pharmacy as well. So let's see what he's saying.